Now that we've taken a look at the makeup of the skeletal muscle, we can take a look at the actual contraction process itself. And so the contraction involves the movement of our thin and our thick filaments across one another. And so this becomes our sliding filament model or our sliding filament theory. And with it, we have the idea that both myosin and actin are going to interact with one another. And myosin and the actin are the myosin will attach to the actin. And if we have a sarcomere here, and those are our Z discs, the Z discs are going to come in towards one another, and we're going to start to approximate them towards the center. So our thick filaments in the center are going to be pulling our thin filaments, and those thin filaments are going to start to move in towards the center of our sarcomere. And so they're going to start to overlap one another. The overall length of the sarcomere becomes shorter, and it's just simply an overlap of our thin and thick filaments that is occurring. And so the thick filaments and thin filaments are overlapping one another, but they're not actually changing the length of those filaments. And so the filaments themselves are not becoming kind of bound up or twisted up or anything like that. They're just simply sliding past one another. So here we have a comparison of a sarcomere when we have a relaxed muscle, a partially contracted muscle, and a fully contracted muscle. And we can see that on a scanning electron micrograph here, that as we go from a relaxed muscle to a fully contracted muscle, that the sarcomere itself is shortening. And so it's going from essentially the end of the Z disc there to there to there. And there's this general shortening of the muscle as it is occurring. The actual filaments themselves are simply sliding past one another. And so again, here we can see our thick and our thin filaments changing in size, the thin filaments becoming smaller and smaller as the space that is there. So the space that we would normally have in our I band is shrinking as that time goes by. And so we can see that the I band and the H zone eventually disappear in that fully contracted muscle. And so essentially those two areas have become completely gone, and all that we're left with in terms of those muscles is the space that is just simply the thick filament, and all of it is essentially overlapped. On the scanning electron micrograph, certainly we can see a little bit of the I band left there, as it's maybe not at its full maximum contraction. So muscle contraction, how does this actually occur? How are we getting to the point where we're going to have a contraction of the muscle itself? Well, when it comes to skeletal muscle has to be told when to contract. So we're going to need to have that muscle have a signal come down from a nerve. And so we're going to have a nerve impulse or an action potential traveling down our neuron here. And as it comes down there and it gets to the axon terminal there, to the end bulb, it's going to essentially transfer that action potential, transfer the signal from the neuron over to the muscle. And so that electrical potential, in a sense, is going to jump from the one cell to the next. But it's going to not just simply pass through. It's not going to move through like having two wires touch one another and the electricity just simply crosses across. We're going to have to utilize a process where we're going to release our chemical, our neurotransmitter, from the neuron over to the muscle. And where this happens is what we call the neuromuscular junction. And so that junction between the actual neuron and that of the muscle is our neuromuscular junction. In between that, we have a little synaptic cleft, so we have a little bit of space that is there, and that's where we're going to essentially transfer our signal in the form of chemicals, which are then going to come over and bind to receptors as they move across that gap that is there. So at that neuromuscular junction, we have the action potential ending here in the end bulb. And as it comes to the end bulb, this is going to cause voltage-gated calcium channels to open up, and calcium is going to rush to the inside of the cell. As calcium rushes into the inside of the cell, this is going to cause vesicles containing a chemical called acetylcholine to move to the end of the end bulb and become exocytosed. So now all of our acetylcholine is moving out into that synapse, out into the synaptic cleft there. And as it binds to the receptors, that then triggers a muscle action potential. So now that action potential is going to travel across the cells away from that neuromuscular junction. And so it's going to travel across and out away from there and move along the surface of the cell itself. As it's traveling along the surface of the cell, our sarcolemma here, it's going to continue to move down those tubes that we had in the cells called our T-tubules. And so that action potential where we're changing the inside of the cell from being, can't really see that there, from being negative on the inside of the cell, all of a sudden those are going to become positive. So we've changed the voltage that is in those. And as that does so, it's moving along the surface of the cell and causing that to occur even down in the T-tubules. The T-tubules are getting this signal, in a sense, this message deep into the inside of the cell where it's next to the myofibrils. And so those myofibrils are then able to start to receive the calcium that's going to get released from our sarcoplasmic reticulum. So the T-tubule is the beige part here. And then all of this kind of network, this web, is the sarcoplasmic reticulum. And the sarcoplasmic reticulum is holding all of the calcium for the cell. So we've got a huge amount of calcium stored inside there. And when that voltage change comes along, that then causes voltage-gated calcium channels to open up, and calcium is going to rush into the inside of the cell. Now it can start to move over to the proteins that are inside our myofilaments. So that calcium is released from the sarcoplasmic reticulum because our voltage has come down along the sarcolemma, down into the T-tubules, and it's reached these sarcoplasmic reticulum. The sarcoplasmic reticulum have voltage-gated calcium channels on them, and when the voltage goes from being negative to positive, those voltage-gated channels open up. Calcium now diffuses into the cytoplasm. It's leaking into the cytoplasm in huge amounts. Very, very large quantities of calcium are being released into the inside of the cell. This then allows the calcium to go out and find troponin. So it's going to go and find troponin on our thin filaments. So the thin filaments are going to bind up that calcium. And when that happens, that's going to allow the myosin head to be able to reach the binding sites on our actin. So our thin filament is going to be bound up by our thick filament, and then we can start to go through that sliding filament process. So the contraction cycle itself begins with calcium entering into the cytoplasm. So it's coming into our sarcoplasm here. As it comes in, it's going to bind to troponin, this little protein here. Troponin, once it binds calcium, changes the shape of troponin, and in the process, it's basically going to drag tropomyosin, the kind of teal one there, it's going to drag that out of the way. And as it does so, it then exposes these binding sites. Now we have binding sites exposed on all of our actin. The myosin head here is lying in wait. It's been energized by ATP, and that ATP has already been broken down into ADP and inorganic phosphate. And so it's already energized. It's in a sense kind of like cocking a gun. It's pulled the hammer back and it's ready to go. As soon as that binding site is available, the myosin head is going to, in a sense, reach up, and it's going to form what we call a cross bridge. And so that cross bridge required ATP in the first place. And so we, in a sense, this is kind of a situation kind of like the chicken before the egg, which has to come first. And so the ATP was there in order to set this process in motion. It already energized our myosin head. So as soon as it's there, that cross bridge forms. It's going to go through its power stroke, and it's going to pull on the actin in towards the M line would be over here. So it's pulling it in towards the center of the sarcomere. And now, in a sense, the myosin is stuck to the actin. And so the myosin is stuck there to the actin. And in order to get it off of there, we need new ATP. So we need to have some new ATP come along. New ATP comes in there, and it's going to weaken the bond between the myosin head and the actin binding site. And as it does so, it now has it released there. We're going to hydrolyze the ATP. We're going to move from ATP into ADP and inorganic phosphate. And now that myosin head is recocked. We pulled the actin in towards the center. Now we're going to go through a process where now that the actin is pulled in, if a binding site is still available and this myosin is reset, it's recocked, it's going to instantly be able to bind onto that binding site, and then it's going to go through the same process again. And it's going to keep on going on and on and on as long as we have adequate amounts of calcium and adequate amounts of ATP available. As long as those two are there, we'll have the ability to continue this process along. And so the cycle keeps on repeating, and we keep on going around and around and around as long as primarily calcium is present. And so as long as calcium is keeping those binding sites open, as long as it's constantly bound there, we're going to continue that contraction itself. And the other thing then is, as long as we don't run out of ATP. So we've got to keep adequate amounts of energy available for this contraction process to occur. And so calcium, ATP required for muscle to contract. If we start to decrease the amount of calcium that's there or decrease the amount of ATP that's there, the muscle is going to start to fatigue. It's going to start to lose its ability to continue to hold that contraction there. And most often the thing that we lose is the ATP. We start to decrease our ability to make enough ATP to supply the muscle with all the energy that it's needed. Not only is ATP required for the myosin heads and recocking of the myosin head or re-energizing of the myosin head, it's also required for our calcium pumps. And so calcium pumps over here are required to get the calcium out of the sarcoplasm and back into the sarcoplasmic reticulum. So we started out with voltage-gated calcium channels. Those voltage-gated calcium channels opened up our calcium release channel, and calcium started to leak into the inside of the cell. That then made the calcium bind to troponin. It changed the shape of the troponin, exposed all our little binding sites here, all the little black dots, and myosin was able to reach up and grab on. So now we've gone from a relaxed sarcomere to a contracted sarcomere. As long as there's plenty of calcium around, as long as there's enough ability to have that calcium there and enough ATP, we'll keep on doing that. Calcium then, once we take away the signal and the cell itself stops having this process of continuing the action potential, calcium pumps are going to pump that calcium back in the sarcoplasmic reticulum. We're then going to be able to hold it in there by binding it to a protein called calsequestrin. So kind of overall, as far as that process goes for our contraction, we start out with an action potential. That action potential comes along, and we have the action potential travel to the end bulbs of the neuron. This causes voltage-gated calcium channels to open up and causes the release of acetylcholine into the cell. From there, we have acetylcholine binding to sodium-gated channels on the muscle. Those open up, sodium rushes in, and we have a muscle action potential that travels over the sarcolemma, down the T tubules to our voltage-gated calcium channels in the sarcoplasmic reticulum. Those calcium release channels open up, calcium comes into the inside of the cytoplasm, floods the inside, it binds the troponin, changes the shape of that troponin, moves tropomyosin out of the way, and now our myosin binding sites are exposed. Myosin can form a cross bridge with the actin. Once it forms that cross bridge, it can then go through its power stroke, pull it towards the center. New ATP is going to come along. That new ATP is going to come along, and it's going to weaken the bond between the actin and the myosin. That's going to cause the myosin to detach. ATP can then be hydrolyzed. We can reset the myosin head, and it's now ready to go all over again. And as long as that calcium is still in the sarcoplasm, we're going to get more contraction. But if it's been taken away from there, that calcium then gets pumped back into the sarcoplasmic reticulum via calcium pumps. And then we're ready, and the muscle is ready and set for the process of contraction the next time we want that muscle to contract. And we'll continue on from there next time.